My name is Sourdive. I am one of the two founders of Cellectis, a company that was incepted about 10 meters away from here, almost 25 years ago. It's very moving for me to be back at the Pasteur Institute, where I was trained as a virologist. I would like to thank you very much for giving me this opportunity. In the next few slides, I'll share with you some highlights of our journey and how we came about fulfilling a vision we had, enabling a couple of revolutions that really are leading to making live drugs, not just therapies, but really drugs. Since we're a listed company and I will be making some forward-looking statements, please refer to our SEC filings. I like to use this quote by Schopenhauer because it illustrates many occasions we went through where thinking out of the box, people were telling us, oh, what you're trying to do is ridiculous. And starting to see it happen, then they said, oh, this is very dangerous. And of course, when it happened, everybody's saying it was obvious. You'll get some of it in the presentation. In essence, when we founded the company with Henri Choulica, just next door, we had this vision, and we started by looking back at what happened to chemistry at the beginning of the 20th century. Chemistry was not unified. There were different fields working on their side until a point where this capability to assemble atoms were congealed into these large players able to deploy that know-how in different fields of applications, including pharma and the first drugs. We thought that biology was about to undergo a very similar process. We had seen what the concepts were, a cell, a protein, a gene, even understood the flow of information from the genome all the way to the phenotype or the behavior of a biological system. We even understood how a few genes could work together. 1965, operon lactose here, very famous place. And by the end of the 90s, after a massive automation of molecular biology, came the first draft of the human genome. We thought there was a tipping point there where it would become possible to rationally rewrite portions of the genome, portion of the program itself, so that we would actually be able to impact the phenotype, measure what it does, go back, change something else reproducibly, measure again, and so forth, and recapitulate the circle that engineers like to practice very much in the development. And of course, that ability to operate on DNA, which is basically the central molecule for the genome of just about everything that's alive, say for some viruses, and that would enable players to deploy this capability to make engineered organisms serve the needs in many fields of application, again, including in pharma, having living drugs. So to fulfill that vision, the first thing we embarked on was to develop gene editing. And we were founded as the first gene editing company. See how we could potentially modify, rewrite portions of chromosomes in living cells and organisms. We started using naturally occurring molecular scissors that do this in nature in small organisms, meganucleases, very hard to engineer, very complex. Later on, we switched to other backbones and used just about everything that was invented since then. Talon, very convenient because they're modular and they wrap around DNA and you can more easily engineer them. More recently came the CRISPR and other avatars, but eventually what we decided was to focus on Talon because to this date they're the most precise, the most accurate and the safest technology to go for clinical applications. Others can be used for very good research, but if you want to go with something very safe, very accurate, you want to use Talon. You can strike within 6 base pairs of any chosen nucleotide in any gene, in any genome of any species. You can work on methylated DNA, unmethylated DNA selectively, work in organelles and so forth. And essentially, we developed that through some years of work into a very solid industrial platform, enabling us to modify, insert, inactivate, replace genes in cells, and even have some new modalities with base editing where we can literally change individual nucleotides at will. So that part being done, and that took years, we actually put it in an industrial setting, being able to deploy it in GMP, i.e. in the pharmaceutical manufacturing. Then we said, okay, if we want to fulfill the mission and the vision, we have to make sure cells become pharmaceuticals, and to that there's 2 revolutions they need to undergo. The first one, we have to make them industrial. They have to be robust, reproducible, characterized. The second thing, we need to make them smart. Ask cells, actually program cells, to make things that normal cells cannot do, not even small molecules or proteins. And so the first application we decided to go for actually came from the field of immunotherapy. A while ago, people had noticed that you can find on the surface of cancer cells some molecular signatures, proteins, receptors, what have you, that are abnormally present, they're abnormally abundant actually. And they've used that set of tumor-associated antigen, as they call them, to redirect the immune system of the patient against its tumors, trying to trigger an immune attack, an acute immune attack like we would have against a virus. They started doing this with antibodies directed to these tumor antigens. They use antibody-drug conjugate, where they basically merge a toxin to the antibody. That's a bit more efficient. They even tried to find the best toxin there is, which is actually killer cells from your own immune system, cells that do specialize in killing back cells. So these are T cells. And they use these sort of dual antibody system bispecific that drag the T cells directly to the tumor to tell it what to kill. And the natural evolution of this continuum came when chimeric antigen receptors were invented, where basically you ask T cells to directly express a new kind of receptor with the same antibody, still the same antibody, but having some signaling system so that this cell is now specific and can recognize directly the tumor as a foreign body and attack it. Now, the three first modalities on the left that you see are all molecular. So it's about developing proteins. The latter is a cell, and that changes everything. Because we know we cannot take cells from just anybody and give them to anybody else. There is a donor-receiver compatibility barrier that has ruled the world of grafts forever. And this is especially true with T cells, because of course, if you take two unmatched individuals and they exchange T cells, the first thing you'll see is the T cells being rejected by the immune system of the receiver. But since the T cells are themselves effector of such rejection, they themselves can start rejecting their environment and attack the healthy tissues of the patient receiving them, leading to what is called graft versus host disease, a very serious condition. For that reason, the first instantiation of CAR-T cells was essentially an autograft, where cells were taken from diseased people who were still fit enough to have valent and valid white blood cells. These cells were taken from the body. They were modified in that material. Genetic material was added to these cells so that they could express this new chimeric antigen, this CAR, that would recognize a specific tumor-associated antigen that would happen to be abundantly present on the surface of their cancer. Cells would be grown a bit, and then it would be infused back into the patients maybe a month later. And that was really the first instantiation, autologous CARs. So CARs, as we've been practicing them, essentially are grafts. They're not a product. They're essentially a point-of-care activity. And this is precisely what we wanted to change. That's the vision we had, to change that. Because when you look at the way cell therapy has grown for decades in the world of graft, makes you want to transition them into the world of off-the-shelf pharmaceutical products. Grafts are bespoke. You have to make one for each patient. They cannot scale. They're highly variable. Actually, you don't even define the product by what it is. You define the product by the process. You say, I've done this with the product, therefore, I can claim I'm injecting the same thing with patients, which is not true. We have the saying, the process is the product. We want to go to a completely different world where we mass produce something that is universal, can be given to anybody, that is robust, consistent, batch after batch after batch. You always produce the same thing. And it is the very characteristics of the product that define it. So the product is the product. In the case of CAR-T, yes, it works. In the case of CAR-T, that meant going for a completely different setting where we would start from healthy donors, people who are just like you and me. Actually, not me, because I'm too old, and healthy in the US means young. I'll come back to that. But essentially, healthy donors that would donate their white blood cells. And these cells would, of course, undergo somewhat of a similar procedure, getting genetic material to actually be able to express the CAR of interest. But on top of that, we would use what we had taken years to develop, which is gene editing, to modify different genes in those cells to break that compatibility barrier and make these cells universal, and be able to make a batch that would treat hundreds of patients out of a fraction of a single donation of a single donor. The scenario we wanted to implement is the following. So in an ordinary patient, cancer cells that thrive are usually evading the immune system. And the concept of the CAR is that because it's going to recognize a tumor-associated antigen, it's going to be able to make the cells react upon contact with the tumor cells and release cytotoxic molecules that kill the cancer cells. The autologous approach that I've showed you about means making such a product for each individual patient. In the allogeneic setting, it's very different. You don't start from disease material. You start from healthy cells. And you have to, of course, add this genetic material to make the CAR, but there's also the caveat of these cells being able to recognize non-self, and yet it's something you want to eliminate. So we use TALEN, these proteins that wrap around DNA that are very accurate. And in this case, what we wanted to do was to inactivate a gene so that it will no longer code for these surface proteins that are undesirable in our case. And doing so massively, reproducibly, in a very robust context in pharmaceutical manufacturing, we are able to have these allogeneic CAR-T cells recapitulate the immune attack on the tumor cells that we want to have taken place. So that's really the scenario that we were trying to develop. And it works the same, basically, looking at the tumor-associated antigen, releasing perforin, granzyme, and so forth, and killing tumor cells. So there's an element of complexity that plays here. We're not talking about molecules. We're not even talking about proteins or something of the kind, or complexes. We're talking about cells, and cells are known to be very complex objects. And looking at the metrics that we have to describe them, they're also extremely variable. Now, if I take the cells of the people in this room and I look at them with the metrics we have available today, I'm going to see a very variable set of pictures. I mean, it's going to be super dispersed. None of your blood looks alike. And if you come back 6 months from today, it's going to have change again. We keep changing. So there's nothing really that looks like a cell, like another cell. It's very, very complex. So how do we tame this variability and make sure we deliver the same product at the end? That was the first challenge. And to address this challenge, we took this process that starts from a healthy donor leukopak, i.e. a bag of white blood cells, and that goes all the way down to hundreds of doses, and we sliced it into different steps, what we call them unit operations, where we transduce the cells, we purify the cells, or we freeze the cells, or every of these steps has been individually optimized, which means optimization of hundreds of parameters: temperature, duration, concentration, what have you. And this work has been done over years, and so that we had these optimized unit operations ready, sort of off the shelf, that we can then combine together for making a new process every time we want to make a new product or a new version of a product. And while the beginnings were tough, I mean, this is real data. We were getting productions that were very dispersed and not very reproducible about a decade ago. It was very tough. But over time, mastering and controlling and optimizing every of these steps, we're able to have very reproducible results, very consistent behavior, and also very consistent markers and product characteristics in the end within very narrow ranges. The time it took us to do the very first processes was very long, was more than 2 years. Now it's down to a few months. Within 3-4 months, we can make an industrial process for one of these sophisticated products very quickly because of modularity. But the truth is not just about modularity. It's also about being exhaustive and being systematic. And the platform we put in place in our premises in Paris, to this date, have completed more than 10,000 runs, over 24 different processes to this date. There's no secret sauce. It's about doing it, and doing it again until you do it perfectly. And you understand every bit of the process. That is really important. So this platform has been able to feed a rather dense pipeline. The first wave of products that were made with this platform are on the bottom, and they were licensed, and they're now being further developed by our friends at Servier and Allogene. And the second wave is on the top, and those are developed by ourselves in acute lymphoblastic leukemia, acute myeloid leukemia, and non-Hodgkin's lymphoma, where we target 2 antigens at the same time, CD20 and CD22. So I won't give you all the details of the clinical results, but after almost 300 patients, what we can tell is that first, we were able to successfully break the donor-receiver compatibility barrier, no issues with GVSD, etc. We were even able to tweak a window of time during which the cells that we infuse can expand, can do their job, kill the tumor before they get rejected by playing with some other knockouts. in terms of performance, when we look at persistence, but more importantly, safety, efficacy, when looking at the same patient populations, we're basically on par with the autologous therapies targeting the same antigens. So that meant that after a first phase where the company really worked on, okay, let's be able to engineer the genome, and then first step, let's make these biological objects real industrial objects, reproducible, robust, drugs, came the second step. Okay, now let's make them smart. Because in fact, what we recognize is that all we're trying to do is to program a particular scenario in the genome of these cells that they will then execute once they're infused into patients. So it's travel, find the tumor, talk to the immune system, kill, call for help. All these things are essentially programmed, and these small autonomous living drones are basically sent into the body, and off they go, and they have to execute this and react properly. Of course, the scenario is likely not to be gotten perfect time right, so there will be tweaking, there will be adjustments, there will be need to look at different features and how we can do that. That's why we call them smart cells. First example that we discussed broadly in a paper recently is called UCART-MUC1, so it's one of these allogeneic CAR-T cell products. It's targeting mucin-1, an antigen associated to known solid tumors, including triple-negative breast cancer. And it has many features to start being able to perform some tasks, a simplified scenario. And let me just walk you through them. The first thing is that this product should be able to be given to anybody. So it is essentially void of its own receptor. It's blind. It can only recognize the tumor. That's basically breaking the donor-receiver barrier. Second, we would like very much this product to be able to travel quite some time in the body and hunt down the tumor wherever it hides. It's not liquid tumors we're talking about. So we actually replace the gene by another, and thereby avoiding the two major mechanisms by which these T cells will be eventually rejected from the patient. So basically, the message is go stealth and evade rejection. The third thing, we'd like these cells, upon encounter with a tumor, not to be subject to the defense mechanism of the solid tumors. We know these solid tumors turn off the immune attacks against them. They have a very immunosuppressive microenvironment, and they keep sending signal to the cells by telling them, leave me alone, don't attack. Two of these major signals are depicted here, hormones like TGF-beta, but also cell-cell contact with the checkpoint and the PD-1, PD-L1 interaction. So what we did is take these one out and just say, these cells should be insensitive to these tumor countermeasures. And actually, instead of expressing those things, we would like very much the cell, on the contrary, to orchestrate a different scenario in the tumor and start an inflammation. So instead of expressing these genes that would make it susceptible to being turned off, we like the cells to secrete a hormone that creates an inflammation, IL-12 and interleukin, and be able to change the scenario. That's a very simple example. Now, of course, when you try it in vitro and see if it works, when you expose and re-expose those cells to tumors, you see that they do express the IL-12 only upon exposure. That's pretty reassuring. It actually helps them control the tumors as well. The more features you add, the better the cells are at controlling an abundance of tumor cells that they're exposed to. That actually translates in animal models. If we take mice and give them a very aggressive version of the tumor that will naturally spread into the animal quickly, taking a version of the product where there's only a CAR, does something, but not so much. If you, on top of that, make the cells insensitive to the countermeasures of the tumor, PD-1, knockout, everything, slightly better. But if you give all the features, including the ability to create some inflammation, then you're much better off and you have a much better results, which actually translates in the survival of the animals. So in essence, we've been able to depict a bit how we program the cellular drones and how it is critical to go quickly from one version to another. Speed is of the essence in this case. And if you take an analogy with electronics, actually on the left you have the first transistor, Bell Labs 9047. I think we are somewhere here with UCART-MUC1, and of course we would love to go further out and have far more integrated, far more sophisticated engineering and programming of these cells. So to be able to do that, to go very quickly from the patient concept, from the design of the cell to delivering a product, we made the decision to re-internalize end-to-end manufacturing of all the elements to make a UCART. It takes a lot of them. It takes 1,000 elements to make one new CAR-T, some of which are custom-made. There are viral vectors, there are messenger RNAs, which themselves use DNA that are actually made custom. So if you actually coordinate this constellation of subcontractors that you can rely on to do that, it's going to take you years every time you want to make a new product or a new version or tweak anything. We compress that to months. So that's really how we got organized to be able to fulfill the vision. We said, no, we want to be able to do versioning. We want to be able to go there because that's the way we will defeat cancer. It's not by sitting around and taking years to change our mind. So also what's interesting is every time we have a new feature, resisting PD-1 or secreting IL-12, these are compounds that we can actually assemble then. And the space of accessible genomic circuitry grows exponentially. The more you have those, the more you can play with them. So we deployed in a setting over 3 sites. We have one site in New York City where the teams design and conceive the scenarios we want to implement to actually attack tumor cells, and that's the way they actually program and think about how to modify genes. It's also the platform. They use a bit of AI to design the molecular scissors, the talent I told you about. Then this is sent to Paris. We have a huge facility here where people do the process development. The 10,000 run team is there, but also we manufacture all the gene editing components, the vectors, the DNA, RNA, and all of that is then sent to a third facility in Raleigh, North Carolina, where the final product is made and delivered for the world. So this allows us to do much more than we can actually prosecute ourselves and develop ourselves. So we've been prone to make a lot of collaborations. I mentioned the ones on the T cells, the CAR T cells with Servier and Allogene. We did some on tumor-infiltrating lymphocytes with Iovance. We also treat at Help Primera, treat genetic diseases that are in organelles. And most interesting, and recently, we've entered into a strategic partnership with AstraZeneca, which is very exciting because it's not just about oncology, it's also about immunology and rare diseases. Very comprehensive and very exciting collaboration. So to wrap it up, what we see now is that the challenge is shifting from being able to technologically modify cells into knowing what to change in the cells and knowing what to program. What is the right scenario? What do we understand of the interplay between the cells, the body, and the disease? And in the case of cancer, there's a tumor, but there are other diseases where it's different. We have to understand the biology, understand how we could make better, how we could change that, and how we can unleash the full power of these smart cells, asking them to do what normal cells cannot do, let alone small molecules of protein. And that's the way these smart cells will take their part in the pharmaceutical arsenal and address the high unmet need of still many, many patients. And with that, I'll thank you and take questions.
Thank you so much, David. This is mind-blowing. When I see genome engineering, synthetic biology, and manufacturing performance, this is science for the industry. Bravo. We have time for Q&A. I have one.
Yes.
And maybe it's a naive one, so bear with me. You never mention for manufacturing scale up or scale out or the scalability. We know that for biologic, that's a real challenge. Here it's more patient targeting. So do you face the same kind of volume and scalability constraints or challenges?
Okay, that's a good question. It's actually precisely an advantage of going allogeneic. From, I would say, 1/5 of a blood donation from patient, we can treat multiple hundreds of patients. That's scalable. We can use all the donation and we can use so many patients. So there's no limit to scalability that we have. We just want to ensure we have very strong, reproducible batch-to-batch consistency. But it's completely different. This is industrial. There is industrial scalability in this case. In the case of autologous CAR-T, it's scale out. And what they have to do is basically have one suite and one batch for every patient. It's more of a point of care activity. It's a graft. Yes. So it's a completely different story.
It's capacity of doing.
So it is naturally scaling in our case. That's really what we do. And the facility we have today, it's not huge. If you were to do autologous, you'd be treating 2 patients at a time. We can treat thousands at a time with that. So it's not the same scale, basically. And it's not the same cost as well. Yeah. That's why cost of goods are so low.
Any questions from the audience over there?
Good morning, Christian Joajo from Estia, Mida, Pyrenees Atlantic. I have some questions. The first one is, have you already applied that technology on human body?
So this technology is already in the clinic, as I showed. There's more than 300 patients, almost 300 patients that have been treated with this technology. And yes, this technology is also being applied directly into the body, but not necessarily through cell therapy. There's also other modalities that are being applied.
Where country? What country?
So today, we have currently trials ongoing in the United States and in Europe, and a few countries of Europe. I don't know if I have them all in mind, but especially continental Europe right now.
The last one, Professor Benjamin Bess from Gustave Roussy said that they encounter some problems with medication. Can that technology help them with the treatment of cancer?
I think it can be a huge game changer because we know that the patients that we are treating have failed everything else. So we have last line patients, people that are in very bad shape. These patients have even failed sometimes the autologous therapies when they had access to it. But it's only a small fraction of patients that have access to these therapies. If you think of the first child we ever treated back in 2015 was an 11-month-old baby. You cannot make a product out of an 11-month baby that is 90% blast. You can't do that. It doesn't work. So there's a whole population, majority of patients still have a high unmet need. And we think that such a product being available on the shelf, not requiring anything from the patient doesn't have to be fit enough or large enough or anything. You just open the fridge, you treat the patient. And I think that could be a huge game changer, especially accessing these very sophisticated cell therapies, cell products.
Thank you.
Any more questions? Reza. Take the mic.
Excellent talk, thank you. Question about the cost of cell gene therapy. Ranges from 300,000 to a new one from Vertex, which is over 2 million. Any thoughts as to how this could be managed in the near future?
Yes. Well, when the cost of a batch is entirely invoiced for one patient, that can be extremely expensive. But that one same batch is invoiced to hundreds of patients. I'll let you do the math.